Thanks, Martin. Thanks for putting the symposium together. I'm going to talk to you today a bit about the use of ultrasound for neuromodulation in humans. Whoops. Okay, so from a general standpoint, ultrasound is an acoustic wave which transports energy, mechanical energy through local vibration of particles in the range of larger than 20 to 25 kilohertz with no net transport of particles themselves. The main mechanisms of ultrasound are either cavitation or radiation force. Cavitation is the interaction of the sound field with a gas body or a bubble, and radiation force is the transfer of momentum from the sound field to the tissue. The main mechanism likely of low-intensity focused ultrasound is likely radiation force, as gas bodies are not typically present in the brain. So the nice advantage of ultrasound is that it can be focused similar to light. And so what we're showing here is that it has very excellent lateral resolution. You can see it's on the millimeter scale there on the left. And the other nice thing about ultrasound is it can also be focused at depth. And so I have a little movie here on the right showing a wave propagation that's focused. So I'll orient you to the figure on the right. So on the vertical axis is pressure. On the x-axis there, that's the face of the transducer. The wave's going to propagate away. And then you can see in the z-axis where the focus is. Sorry. Okay, so now you can see the wave is propagating and has a low amplitude. And then depending on the focal length that you wish, that's where you can have the bulk of the pressure. And in this case, it's roughly 5 centimeters from the source. Oh, that one.
No, this is different.
Okay, thank you. So this is a nice feature of ultrasound, but does ultrasound affect brain activity? And in multiple models, this has been shown to be true. Ultrasound can stimulate the motor cortex in mice. It's been shown to inhibit visual evoked potentials in rabbit and also produce a bold response in rabbit. It's also been shown to affect behavior in monkeys. And so my work looks at the effect of ultrasound in humans. This is our general setup. We have a function generator that produces the waveforms. Pass it to an amplifier, which is attached to the transducer, and then you would place the transducer on the scalp over your desired cortical or subcortical area. What I'm showing on the left is one of our typical pulsing schemes. We don't use continuous wave ultrasound. We pulse it, and so we have a pulse repetition frequency. In this case of 1 kilohertz, we do 500 pulses to give us a duration of stimulation of half a second. We use an acoustic frequency of half a megahertz and a duty cycle of 36%. And so our basic strategy in our initial studies was to look at the effect of ultrasound on the somatosensory cortex, and particularly on the amplitude of somatosensory evoked potentials. And so down on the left, I'm showing you kind of the temporal schematic. So we record surface EEG continuously, and then to elicit the SEPs, we do a median nerve stimulation, which is represented here on time 0, the MN stim. And then we would deliver ultrasound here in blue at a timing relative to the onset of the median nerve stimulation. Under the guise that we wanted to see what does ultrasound do to the amplitude of the SEP if it's delivered while you're generating the SEP. I have a little video down here on the right showing that's kind of a typical transducer that we would use. And if you focus your attention on the surface of the water, you can see how the ultrasound displaces the water in very fine spatial precision. And then we also have, later in the video, we show that it can also translate through a human section skull here. So you can see the water displaced nicely here with good spatial resolution. I hope you can see that. Okay. So what did we find? So in the top left corner is a model of the size of our ultrasound beam relative to cortical geometry. So S1 is the primary somatosensory cortex, or the postfrontal gyrus. The CS is the central sulcus, and M1 is the primary motor cortex. And you can see that the lateral size of the beam is smaller than a human gyrus, so we do get good spatial specificity. We also have a coronal section there in our model, and we've blown it up in the bottom left. And you can see that the beam well approximates and has good spatial resolution with the posterior bank of the central sulcus. And this is important because this is where these somatosensory potentials have been sourced to. So we wanted to see if we focus the ultrasound there, does it affect these potentials that are generated there? So on the right, we have that's a somatosensory evoked potential recorded from electrode site C3. On the y-axis, we have amplitude in microvolts, and then on the x-axis, we have time in milliseconds. The timing of the ultrasound being on is represented in the blue bar. And so we have a sham condition in the blue trace and the ultrasound condition in the red trace. And generally what we found was ultrasound directed at the somatosensory cortex resulted in inhibition of the evoked potentials recorded in that area. So because it has such high spatial specificity, is the effect limited to the size of the beam? And so one of the ways to test this is if you translate the beam off the original spot, do you still get the same effect? So we translated the beam either 1 centimeter anterior to the original spot or 1 centimeter posterior to the original spot and looked at the amplitude of the somatosensory evoked potentials. As you can see on the right, we saw no difference between the red and the blue trace when we translated the beam. In this case, this is the data from the 1 centimeter anterior trace. So from the somatosensory cortex, we wanted to move to the motor cortex, but we have a small little problem in humans. So Dr. Konefegu showed in mice that if you deliver ultrasound to the motor cortex, you get these nice peripheral motor responses. Well, that's not the case yet in humans. We haven't figured out how to do that. So if I want to test how ultrasound affects the motor cortex in humans, how do I do that? So we decided that we would pair ultrasound with TMS. And so what we show up here in the top left is a typical figure 8 TMS coil with a neuronavigation system attached to it. And we developed a custom-made transducer that we could attach to the TMS coil, and so we can deliver concurrent and concentric ultrasound with TMS. And so the purpose of this is so that we can use TMS to evoke a motor evoked potential, and then we can see what does ultrasound do to the amplitude of that motor evoked potential. So the first thing that we did was looked at TMS recruitment curves. So on the left here, we have some recruitment curves. That's MEP amplitude on the y-axis and percent TMS output on the x-axis. So you slowly ramp up the TMS, and the amplitude gets bigger, and we want to see you do this with and without ultrasound. So we have the sham condition in black, and then we have the TMS with the ultrasound in gray. And as you can see, when you deliver ultrasound concurrent with TMS, it results in an inhibition of the amplitude of these MEP potentials. So this is data somewhat similar to what I showed in the somatosensory cortex. If ultrasound is going to be demonstrated to have some kind of therapeutic effect, it'd also be nice to know if it has any lasting effect outside the time of stimulation. And so we thought that this pairing of TMS with ultrasound is highly advantageous to test the temporal effects of ultrasound. And so what we did was we temporally offset the timing of ultrasound with TMS. So you can deliver concurrently and see what it does, or you can deliver ultrasound and then test TMS at different time points later on. And that's what we're showing here on the right side. So on the y-axis is a normalized MEP amplitude, so BL is baseline, that's 1. And then we have these different temporal offsets on the x-axis. So the negative 100 refers to delivering ultrasound concurrent with the TMS. And then you do TMS at different time points post-offset of ultrasound. 100 milliseconds after, 200 milliseconds after, 500, all the way up to 10 seconds. And so in this case, we're delivering a 500 millisecond duration of ultrasound. And so what we found is that the bar, the negative 100 bar, we show the inhibition, so this is when they're delivered at the same time. And then it looks like we do get sustained inhibition, at least in this case, out to 500 milliseconds, and then the MEP recovers. So we seem to have, in this case, we just tested a duration of 500 milliseconds of ultrasound, and it looks like it lasts roughly 500 milliseconds, but we're continuing to test different durations and different parameters to see how long we can get the effect to last. So we also looked at the effect of ultrasound on paired pulse TMS. Paired pulse TMS is a technique to either induce inhibition or excitation in the motor cortex, and also gives a window into different motor microcircuitry within the motor cortex. And so what we have on the top is normalized MEP amplitude in the y-axis, and then we have these different ISIs on the x-axis. And so if you deliver 2 TMS pulses in close temporal proximity on the left, you can see that you get inhibition, and if they have a longer temporal offset, you get facilitation. And so we have our sham data without the ultrasound in the black line, and then we have with ultrasound in the gray line. And so we didn't find ultrasound to have any effect on the inhibition at the short ISIs, but we did find that ultrasound inhibited the facilitation from paired pulse TMS. And then the group data is represented in the bars on the bottom there. Okay, so we're going to switch to the use of ultrasound for potentially for subcortical stimulation. So we mentioned that you can focus it at depth, and so we wanted to test this in humans, and we decided to see if we could use ultrasound to non-invasively modulate the thalamus. And we chose the thalamus because there is a synapse in the thalamus, the VPL, shown up there in the left in green, that is part of the dorsal column medial meniscal system, and that you can measure this synapse from non-invasive from surface EEG as this P14 somatosensory evoked potential. So the theory was that we can point the transducer at this part of the thalamus, and then we can measure that activity in the thalamus non-invasively from surface EEG. So up top, we have one of our acoustic models that takes into consideration the individual subject's MRI and also their CT scan. They are overlaid there. And you can see in the center, in the sagittal plane, we have very excellent lateral resolution. And you can see in the coronal and also in the transverse plane that we have fairly good axial resolution as well. We can certainly target the thalamus. It's debatable whether we can target anything within the thalamus, but the point is that we can get energy to the thalamus non-invasively using ultrasound. And so on the bottom, I'm showing somatosensory evoked potentials again. We recorded from 5 sites. And so on the bar graph there on the left, you can see the ultrasound condition in red and the sham condition in blue. And we did see an inhibition of this potential as a result of ultrasound as compared to sham. We also showed in the SEP traces, those gray bars represent statistically significant differences as well. And some of these potentials have been localized to other areas of the somatosensory cortex. And so it looks like this effect, when you stimulate the thalamus, may translate also to cortical areas. But you may say that maybe EEG isn't probably the best modality to look at the spatial specificity of ultrasound, and I would agree with you. So we've also been looking at the combination of ultrasound with MRI. And so Sun-Shik Yu's group showed very nicely in rabbits that you can induce a bold response as a result of ultrasound. And so we wanted to see if we could do this in humans. And so we have a transducer, in this case, over the motor cortex. We did this in several subjects. I'm only showing 3 here where we did get a fairly specific, nicely isolated bold response. But we have several subjects where this wasn't possible. So this is turning out to be highly variable for reasons that we're not too sure of right now. And so I did this work with Jamie Tyler at Virginia Tech. And then I got a job at Minnesota, and I wanted to do it in Minnesota, but they wouldn't let me do ultrasound on the 3T scanner for safety reasons and stuff, and I was like, oh, nuts. But the safety committee allowed me to do it at 7T, which is interesting. So now we're doing this in 7T, and I was like, that's great. So we have improved resolution, and we have improved signal to noise, but we still have the same problems. I'm showing here one subject where we are able to show very discrete bold responses. Those are 2 trials. The blue does not represent negativity. It's just a separate trial. So we can get bold responses that approximate well to where the beam goes, but it's highly variable. We don't get in all subjects, and again, we're still working on why this is and trying to optimize it. So instead of looking at, we decided on a new strategy. So it's proving difficult to induce bold all the time. So what happens? Does ultrasound just affect bold? And so we did a different strategy. So we said, well, we'll induce bold through a finger movement, and we'll see what does ultrasound do to that bold response. So what I'm showing here is, you can see in the figure in the bottom right, we have a thumb activation in the primary motor cortex, and I've overlaid the size of our ultrasound beam and just kind of translated it over so you can see the spatial size. And so what's nice is that the resolution of the ultrasound beam is commensurate with the size of an individual finger representation in M1. So this is fantastic. So in theory, we can target individual finger representations within M1 with ultrasound. And so that's what we tried here in this study. So we delivered, people would either move their thumb or their index finger or their middle finger. We found out where their activations were, and then we would put ultrasound on those activations to see if they just affected the thumb region or if it translated to the index region or to the middle finger region. So the data I'm showing on the right is from 5 subjects. They have the sham condition on the left column and the TFUS and the ultrasound condition on the right column. And so interestingly, we did not, this is just the thumb data I'm showing, by the way, as well. So we didn't find any difference in a % signal change as a result of ultrasound. But what we did find was we found volume expansion. And so that's represented down there in the bars. The reasons for this are unclear. We're continuing to do this research. And unfortunately, I don't have a definite conclusion as of yet. Okay, time's running out. So I'll just leave you with the last things that we're doing is we're really interested in the effect. Does anatomy have any effect on ultrasound? So as you may or may not know, electric fields generated by TMS or TDCS are highly susceptible to gyral foldings. Is that the same case with ultrasound? And so we did some fairly rudimentary models where we could place a beam and we could make different layers of cortex, and then we could change the shape of the geometry of the head. And we're pleased to say that regardless of the changing of the geometry of the brain, that this does not affect the beam size at all. So ultrasound is independent of human gyral foldings, which is good. However, the one issue with ultrasound is the skull. So the skull is highly attenuative to acoustic energy. It absorbs it, turns it into heat, can reflect it or refract it, and so this is a big issue. And so we do a lot of modeling, taking into consideration individual skull morphology to see what it does to the beam, to the pressure and to the geometry. And the data I'm showing on the right is just the effect of skull thickness on the x-axis relative to the pressure in the head on the y-axis. And so we put the transducer on two positions on the head. We put it on where the skull is thinnest in the temporal window, and also where the skull is thicker over the parietal bone. And so that's what that's on what the data on the right with the connected lines are. It's one individual with the thin part of their head and the thick part, and we were looking for any kind of relationship between bone thickness and pressure, and we didn't find one. And so this just suggests that you do have to take individual skull morphology into consideration when you are doing ultrasound in humans. So in conclusion, is ultrasound brain stimulation the future? I think it's got a place in neuromodulation. It's got improved spatial resolution to magnetic and electric modalities, and it has the ability to be focused at depth. So that's a big advantage. And the other advantages are pointed out here on the right. This is a recent review paper by Jeremy Tyler that I suggest that you read if you're interested. And with that, I will thank my funding sources in the universities and my lab. And I'd also like to do a shameless promotion that our lab is looking for people who are interested to study ultrasound. And so you can visit the website, give me an email, or we can talk after this. Thanks for your attention.